आइए अब हम प्रोसीजरल स्टेप्स के हवाले से कुछ बात करते हैं दैट आर गोइंग टू बी इन्वॉल्व्ड इन द प्यूरिफिकेशन ऑफ प्रोटीन्स। हम इनको डिफरेंट स्टेप्स में डिस्कस करेंगे। फर्स्ट स्टेप वी आर गोइंग टू टॉक अबाउट इज प्रेपरेशन ऑफ सेल लाइजेट। जैसा कि पहले भी इस हवाले से बात की कि हमारे पास सेल लाइजेट या सेल एक्सट्रैक्ट बनाने के लिए डिफरेंट मेथड्स हैं। वी कैन यूज़ मैकेनिकल डिस्ट्रप्शन ऑफ द host cell, we can use sonication, use of the sound waves, or we can use the enzymatic digestion method. In this case we are going to talk about use of enzymes to prepare the cell extract. जैसा कि कल्चर के हवाले से बात की थी that we should start with a large volume, that is 1 liter. इसका basic purpose क्या है that in 1 liter cell culture or the bacterial culture we will have sufficient number of bacterial cells expressing our gene of interest. So at the end of this extraction we will end up with sufficient quantity of the protein. So 1 liter volume is big enough which cannot be handled during the subsequent steps. So we have to reduce the volume of this culture medium and we have to separate the bacterial cells from the liquid culture so that we can use them for preparation of cell extract. For that purpose we have to centrifuge the bacterial cell culture at maximum speed or highest speed of 5000 G for 15 minutes at 4 degrees centigrade, and the tubes in which we are going to perform this centrifugation, they should be weighed before starting the centrifugation so that we can find out the weight of bacterial pellet after centrifugation. Once centrifugation is done, the upper supernatant is no more required. So we remove that supernatant. Whatever is left behind is a pellet of bacterial cells in the tubes, centrifuge tubes. So now we have to determine the wet weight of the bacterial pellet. यहाँ पे इसको इस तरह से एग्जाम्पलीफाई किया जा सकता है कि अगर आपके पास सेंट्रीफ्यूज ट्यूब जो है बिफोर स्टार्टिंग सेंट्रीफ्यूगेशन वाज 15 ग्राम्स और सेंट्रीफ्यूगेशन के बाद सुपरनेटेंट डिस्कार्ड करने के बाद उसका वेट किया जाए, वेट इज़ 25 ग्राम्स. So 25 minus 15, the bacterial pellet is around 10 grams. Weight of this bacterial pellet is important. उसकी वजह क्या है कि आगे चल के हम जितने भी decisions करेंगे, how much volume of any enzyme, how much volume of any buffer reagent is going to be used, is going to be dependent upon this weight. So this is a crucial step, that at this stage you determine the wet weight of the bacterial pellet, and once the weight is determined, then we add 3 milliliter of lysis buffer 1 for each 1 gram of bacterial pellet. अगर आपके पास pellet जो है सेंट्रीफ्यूगेशन के बाद 1 ग्राम है तो हम उसमें 3 मिलीलीटर lysis buffer 1 ऐड करेंगे. अगर 2 grams है then we are going to add 6 milliliter of lysis buffer and so on. Once the lysis buffer is added, the next step is resuspension of the pellet. We want that all the bacterial cells are resuspended in the lysis buffer so that each and every bacterial cell in that particular lysis buffer should be exposed externally to the lysis buffer, to its components, and its process of lysis can start. After resuspension of the bacterial pellet, we add 4 microliter of PMSF in the tube, again per gram of E. coli. This volume will be adjusted as per the weight of the E. coli. 80 microliter of lysozyme stock is added, 10 milligram per milliliter. Now this lysozyme is going to perform the enzymatic action on the cell wall and is going to cause the cell wall disruption. इस पूरे mixture containing the PMSF lysozyme in the lysis buffer is going to be stirred for 20 minutes. Is 20 minutes में enzyme will have sufficient time to perform its action, and then we can proceed further towards cell lysis. अब यहाँ पे एक स्टेज क्रिटिकल है और ऑप्शनल है. We should not ignore the presence of proteases. अब प्रोटीएजेस वो एंजाइम्स हैं जो सेल के अंदर प्रेजेंट होते हैं और जो प्रोटीन को डिग्रेड करते हैं. सो अगर हमें अपनी प्रोटीन के हवाले से कॉन्शियस रहना है, उसको इंटैक्ट फॉर्म में लेना है, एट दिस स्टेज वी कैन थिंक अबाउट एंड वी कैन डिसाइड दैट वी शुड ऐड अ कॉकटेल ऑफ प्रोटीएज इनहिबिटर्स. उससे क्या होगा, जैसे ही सेल लाइज होगा, प्रोटीएजेस जो लाइजेट में आएंगे, प्रोटीएजेस आर गोइंग टू बी इनहिबिटेड बाय प्रोटीएज इनहिबिटर्स. अब वो प्रोटीएजेस इनएक्टिव हो गए, वो हमारी किसी भी प्रोटीन को, चाहे वो सेलुलर प्रोटीन है चाहे वो फॉरेन प्रोटीन है, उसको डिग्रेड नहीं कर सकेंगे. While performing this extraction process, stirring continuously is going to help the bacterial cells to be exposed to all the ingredients of the lysis buffer. In the meantime, we are going to add 4 milligram of deoxycholic acid per gram of bacteria. Again, weight is important here. This is going to act as detergent. In the presence of enzyme and detergent, the lysis process keeps on going. After this addition, we store this suspension at 37 degrees centigrade and stir occasionally with a glass rod. Occasionally we can stir it with a glass rod so that the things keeps on mixing up. अब इस इंक्यूबेशन पीरियड में एक स्टेज ऐसी आएगी जब हमें विस्कोसिटी के हवाले से फीलिंग होगी दैट द विस्कोसिटी इज चेंज्ड. When the lysate becomes viscous, that is an indication that cytoplasm has been released in the lysate, and now DNA, protein, RNA, everything is in the lysate. At this stage, we need to add DNase 1 enzyme so that we can get rid of the DNA which is present in the lysate, and then we leave it as it is for next 30 minutes so that the DNase 1 performs its function and the viscosity of the lysate goes back to normal, so it is no more viscous.